nella seduta del 13 dicembre 2004, ha deliberato all'unanimità di conferire la laurea Honoris Causa in Chimica al professor L.H.C. Brant, in considerazione delle tappe significative da lui percorse nel campo della chimica macromolecolare e della chimica dei carboidrati. La facoltà vuole riconoscere il rigore scientifico su cui è basato tutto il suo lavoro e le doti di originalità nello sviluppo di approcci teorici e sperimentali che hanno contribuito notevolmente alla comprensione di fenomeni complessi nel campo dei carboidrati polimerici. Inoltre, la facoltà ha particolarmente apprezzato il ruolo svolto dal professor Brant come docente, le sue qualità didattiche e di disponibilità verso colleghi e studenti che gli sono sempre state unanimemente riconosciute. Infine, la facoltà esprime il massimo compiacimento per il rapporto culturale ed umano che il professor Brant ha avuto con numerosissimi ricercatori a livello locale e nazionale. Per coloro che hanno avuto il privilegio di ricevere da lui insegnamenti, egli ha rappresentato anche un costante punto di riferimento scientifico nelle molte occasioni in cui è stato a Trieste.
Bene, ringrazio professor Ruzier e ora do la parola invece al professor Cesaro che illustrerà le motivazioni anche legate naturalmente al profilo scientifico, culturale e professionale del professor Brant. Prego, professor Cesaro.
Magnifico lettore, cari colleghi, gentili signore e signori. Descrivere la carriera del professor David Brant e tessere le sue lodi rappresenta per me, oltre che un onore, soprattutto una sensazione di profonda e grande gioia e sincera soddisfazione. Lo farò tracciando alcuni elementi biografici del suo ruolo presso l'Università di California, della sua attività scientifica e didattica e del suo rapporto con l'Università di Trieste. Il professor Brant è stato come professore di chimica insieme a Robert Taft, Marjorie Caserio, Harold Moore, Sherry Lawrence ed altri uno dei membri del nucleo fondatore dell'Università di California Irvine, che quest'anno celebra i 40 anni della sua istituzione. Oggi questa università è classificata come una delle migliori a livello nazionale USA e soprattutto per gli studi in chimica. Ad Irvine Brant ha rapidamente ottenuto tutti gli avanzamenti di carriera, diventando full professor nel 1974. Egli ha contribuito attivamente alla formazione e alla vita accademica, ricoprendo diversi ruoli, quelli che in Italia denomineremmo come direttore di dipartimento, preside di facoltà, presidente del Senato accademico. In tempi più recenti, come anche ha menzionato il rettore, al professor Brant è stato affidato il compito di riorganizzare il Department of Education per la gestione di un grande progetto didattico della California del Sud. Le tappe del suo percorso scientifico lo hanno visto passare prima alla Yale University per poi conseguire il dottorato di ricerca in chimica fisica con il professor Alberti all'Università del Wisconsin. Successivamente ha svolto le sue ricerche alla Stanford University dal 62 al 65, collaborando con il professor Paul J. Flory, forse il più prestigioso nome della chimica macromolecolare. Flory ha poi ricevuto il premio Nobel per la chimica nel 1974. Tra i risultati più rilevanti delle sue prime ricerche vanno soprattutto ricordati i lavori memorabili sulla statistica conformazionale di polipeptidi nel periodo 64-66. Nei primi due lavori vengono poste le basi dello studio conformazionale di strutture statisticamente disordinate, dapprima, come rigorosamente chiede l'approccio scientifico, attraverso l'osservazione sperimentale e poi attraverso l'interpretazione teorica dei risultati. Prima l'esperimento e poi la teoria, e questo è il modo naturale di sviluppare la conoscenza scientifica. Forse è questa anche una forma di teoria dell'armonia ritrovata, se mi è concesso di parafrasare lo scrittore Raffaele La Caprica. Questi studi hanno fornito le chiavi di lettura della stabilità conformazionale delle proteine, come è stato poi successivamente verificato da altri ricercatori e come è stato anticipato nel lavoro con Paul Schimmer nel 67, poi riportato su quasi tutti i testi di biofisica e biochimica macromolecolare. Come ha raccontato lui stesso durante un recentissimo simposio all'American Chemical Society a Orlando, il trasferimento da Stanford a Irvine nel 1965 ha catalizzato quella sorta di conversione che poi ha fatto di Brant un pioniere nel campo dei polisaccaridi. L'identificazione di una strada inesplorata e la curiosità scientifica, unita a un calcolato rischio dell'impresa, lo hanno convinto dell'opportunità di questo cambiamento. Nel settore dei carboidrati polimerici e in particolare degli amidi, come acuti ricercatori dell'epoca hanno riconosciuto, era spesso difficile separare le osservazioni scientifiche dal folklore. Così le ricerche di Brant e Irvine sono tutte rivolte a portare su base quantitativa di assoluto rigore scientifico gli studi nel campo dei carboidrati polimerici. In tal senso si muove con autorità nel definire passo dopo passo tutta la struttura di calcolo necessaria per affrontare lo studio delle conformazioni di polisaccaridi, giungendo nel 1975 ad un mirabile lavoro A General Treatment of the Configurational Statistics of Polysaccharides, pubblicato su Macromolecules. Negli stessi anni Brant elaborava un progetto che per la prima volta utilizzava il metodo di simulazione, noto come metodo Monte Carlo, per la descrizione statistica delle catene polisaccharidiche e per la rappresentazione visiva di istantanee di singole catene polimeriche. E fu coniato il termine snapshot, diventato poi gergo comune. Da allora sono apparsi diversi altri lavori, capitoli di libri e annual reviews in cui David Brant, qui negli anni '80, ci ha aggiornato sulle strategie di calcolo con uno stile chiaro, comprensibile e mai superfluo, sempre alla ricerca del miglior risultato con il minor numero di assunzioni. L'innovazione, unita sempre alla corretta interpretazione, è stata una dominante nei lavori di Brant. Mi piace particolarmente citare un caso, precursore di tipiche indagini nel campo delle nanoscienze. Non appena le modifiche alla tecnica di microscopia a forza atomica, la non-contact Atomic Force Microscopy, lo hanno permesso, ecco che i primi lavori nel campo dei polisaccaridi delucidano aspetti inconsueti, quali quelli della formazione di polimeri ciclici, a cui fa seguito tutta la trattazione termodinamica statistica a giustificarne la non casualità. Tuttavia il lettore attento troverà in questi suoi lavori anche un rigoroso uso delle parole e dei concetti, che ci fanno comprendere quanto forte sia stato in Brant il senso della correttezza intellettuale, di quanto cioè potesse essere scientificamente affermato rispetto a quanto dovesse essere considerata mera speculazione. Non parlerò di altri risultati sulla dinamica locale che pure ci hanno visti coinvolti, ma voglio sottolineare che per i suoi lavori egli ha ricevuto nel 2001 la medaglia Larsson-Sager dell'Accademia norvegese delle Scienze e Tecnologie. Se è conclamato il valore scientifico delle sue attività di ricerca, è indubbio che la classificazione della chimica Irvine nella top list delle università USA sopra altre prestigiose sedi come Caltech e UCLA è dovuta non solo all'attività scientifica ma anche di quella didattica. Occorre infatti sottolineare l'inscindibile correlazione e l'unità di significato che hanno, e spero non solo per le università americane di prestigio, l'attività di ricerca e l'attività didattica. Che la qualità della didattica non sia un parametro aleatorio, ma un rilevante fattore di merito, è evidente nell'importanza che la sua valutazione ha all'interno del mondo universitario. Il professor Brant per questi meriti è stato più volte insignito di pubblici riconoscimenti a livello nazionale nel 1977, nell'88 e nel 2001. A noi piace ricordare la chiarezza espositiva delle sue lezioni e dei suoi seminari, che evidenziava un'attenta e curata preparazione del materiale didattico, la profonda conoscenza dei contenuti scientifici e delle forme della sua disseminazione, la pacatezza dell'esposizione delle sue tesi, sia che egli insegnasse, come è accaduto e accade negli USA, biofisica che chimica analitica, sia chimica fisica che biopolimeri. E tutto questo con la sua continua e più ampia disponibilità verso gli studenti, che hanno sempre trovato la sua porta aperta ed una sensibilità profonda ad ascoltare e a consigliare. Infine mi sia concesso di motivare il rapporto particolare tra Brant e Trieste, con delle annotazioni personali ma non soggettive. Occorre andare indietro, alla fine degli anni '60, al periodo iniziale del suo arrivo a Irvine e al suo nuovo interesse per i carboidrati. Ripercorrendo quegli anni non possono ricordare di essere stato affascinato e come me tanti altri dalla saggezza del suo insegnamento, dalla responsabile guida in tutte le occasioni. Di quel periodo mi resta un gran bagaglio culturale di ricordi scientifici e umani e anche la copia di una foto del 67, pubblicata in quella che corrisponde alla guida dello studente di quegli anni. Successivamente ci sono state tante altre occasioni di incontri, dei quali sono soprattutto da menzionare i periodi di studio di Brant a Trieste nell'81, nel 90 quando è stato visiting professor e infine nel 93. Colleghi e studenti dell'Ateneo Triestino hanno avuto la possibilità di ricevere insegnamenti e hanno potuto usufruire di costruttive discussioni e di suggerimenti per il loro lavoro. A Trieste egli è stato sempre accolto con grande calore e profonda stima. L'occasione odierna è appunto la testimonianza di questo vivo legame umano e scientifico. In conclusione, come ho avuto modo di sottolineare in un'altra sede, se la funzione di base di ogni educazione, anche nel più tradizionale significato, è quella di incrementare la probabilità di sopravvivenza di un gruppo di individui, diventa evidente che per quanto ci riguarda questo scopo è stato pienamente raggiunto, in considerazione dell'attiva comunità scientifica che si è sviluppata su quel fertile terreno di conoscenze in tutto il mondo e soprattutto a Trieste. E ne ho avuto testimonianza in questi giorni per le numerose lettere di congratulazione da indirizzare a lui. Di tutto ciò desidero ringraziarti a nome di tutti i colleghi di Trieste.
Grazie. Vorrei invitarvi ora ad alzarvi perché darò lettura. Would you mind to make a few steps forward? Thank you. So, in nome della legge, scusatemi perché è importante anche questo. E noi Domenico Romeo, magnifico rettore dell'Università degli Studi di Trieste, visto il testo unico delle leggi sull'istruzione superiore, sul delibera del Consiglio della Facoltà di Scienze Matematiche e Fisiche Naturali del 13 dicembre 2004, vista l'approvazione del Ministero dell'Istruzione e dell'Università della Ricerca, conferiamo a David Ames Brant, nato a Kingsport, Tennessee, il giorno 20 settembre 1936, la laurea in chimica ad honorem, in considerazione delle straordinarie doti di ricercatore nel campo della chimica macromolecolare e di riconoscimenti avuti per l'autorevole e originale contributo allo sviluppo della statistica conformazionale di polisaccaridi attraverso il complementare utilizzo di approcci teorici e sperimentali. Io penso che ci sia la toga, pronto.
Professor?
Prego, accomodatevi. Il professor Brant tiene adesso la sua Lectio Doctoralis e il titolo è Versatilità dei polisaccaridi, filamenti, anelli e bacchette. Please, Professor Brant.
Rector Romeo, Dean Guzia, Professor Cesaro, and all of the assembled guests, thank you very much for this honor. I greatly appreciate it. It's a great honor for me to be here, and I very much am pleased to be here. My first connections with the University of Trieste began in 1962 when I was in the laboratory of Paul Flori. I met a gentleman named Vittorio Crescenzi. Vittorio Crescenzi at that time was, I believe, still at the University of Rome, a young professor there. But subsequently, very soon after we both left Stanford University, he moved to Trieste and began a program here, which was one that I began to collaborate with immediately. When I left the Stanford University to begin work at the University of California, as Attilio Tesaro has mentioned, this was a brand new university. The university was started in 1965. I went there. I was one of the first faculty members, and it's the only job I have ever had. I have been there for 40 years. I needed to find some area of research that I could call my own, that was different and new. And while I had been working in the polypeptide area on proteins, I felt that I needed to find something completely different. And so I began to look around for some other biopolymer to study. And I decided to begin work on polysaccharides. And I've been working on polysaccharides the entire time I've been at the University of California, Irvine. The title of my talk here says that they are versatile. They are versatile because they can take very many different shapes. And what I want to talk about today, I'm not going to try to talk about everything I've done, but I do want to talk about the variety of shapes that polysaccharides can take, because it's through that variety of shapes that their physical properties, their biological properties, and their commercial properties are realized. So let's begin by talking about what we mean by ropes, rings, and rods. I think ropes is properly translated into Italian as filamenti. So long strings or ropes which are very flexible. Rings, of course, anelli, another shape that polysaccharides take, and rods, I think the correct translation is bacchette. So I'm going to talk about each of those three categories. I'm going to talk relatively briefly about the ropes and relatively briefly about the rings. I'll spend most of the time talking about the rods, the rod-like polysaccharides, which I think are some of the most interesting. So I understand that this is a very general audience, that many of you are not chemists. I'll try to be rather general in my approach to this. So what is it about that characterizes ropes? I will call them also random coils. A polymer, of course, is a chain of many units. And the characteristic of these random coils is that the links between the units are very flexible. In contrast to proteins, which realize their function because of the very specific folding that they do. They fold up in a very specific way to generate their biological properties. Polypeptides typically do not fold in the same sense that proteins do and that many nucleic acids do. In fact, many of them can adopt a very wide range of shapes, all of these shapes having nearly the same energy. And that means that at any given time, the molecule is always changing its shape, and that all of the molecules in a sample, every one has a different shape. So we have a tremendous variety of shapes. And so that's the characteristic of the random coil. Now, what we have here, if you look at the slide, you'll see I'm pointing to the picture here. These are a couple of sugars connected together in a particular way. These are glucose. And the connection between them is you can conduct rotations about these two chemical bonds that are linked by a phi and a psi. And you'll see plotted on this graph here the phi and the psi. So this contains the entire range of rotation about this bond, and this is the entire range of rotation about the other bond. And what this diagram intends to display is the relative energy of those various shapes. So there's a relatively small range of these shapes right here in the center, which are quite low energy and therefore accessible. Where you see just plain white here on the diagram, that's very high energy, and the molecule cannot achieve that status. But right here where the X is, and this region around, this is the range in which these two sugars are changing with respect to one another in terms of their shape. Now, this particular connection between two glucose molecules produces polymer chains of a particular shape. And the next slide I'm going to show you is like a photograph of one molecule frozen in time. So here you see a picture of a molecule contained with glucose molecules hooked together or connected in the same way that I showed you before. These circles that you see are just a very symbolic representation of the sugar. So don't think of this is not a detailed description of the structure, but simply written this way so you can see what the trajectory of the chain is. And what you see here is that it's not very orderly, but you do see a tendency for some helicity. And that is a reflection of the way that those two sugars are hooked together. Now, so that's one sort of mode of shape that polysaccharides take. Let's go on to another one. Here, we've hooked the two sugars together in a rather different way. So a sugar here and a sugar here, but now instead of having just two chemical bonds between them, now there are 1, 2, 3. And because there are 3 chemical bonds separating the sugars, there's a great deal of additional flexibility. So because there are 3 bonds, it's not possible to draw a simple diagram. But what I want to illustrate here is that if you examine this bond right here that's labeled omega, that's like moving in this direction. And what this diagram is telling you is that it's very easy to rotate around this bond. So rotations occur without much difficulty, without many hindrances. And that's likely to produce a very flexible chain. And the next diagram shows what a chain like that would look like. And here you see that it's really quite disordered. There's no pattern that you could see. So if I took another photograph a second later and another photograph a second after that, the picture would look quite different. This is a really true random coil. In polymer physics, the idea of a random coil is a very dominant notion. And this particular kind of polysaccharide really comes very close to being a chain with flexible joints between the units. So this is another mode in which sugar chains take a random coil shape or a ropey shape. I'm going to show you just one more example like this. Now this looks very much like the original diagram I showed you, but now the connection between these two sugars is in a different position. So this is a connection between position 1 and position 3. The first one I showed you was a connection between position 1 and position 4. And the thing that makes it clear that these are different is the completely different shape of this energy diagram. This energy diagram now has a completely different shape. Here's the lowest energy shape, but there's a much broader range of possible shapes for this pair of sugars. And so we can anticipate that this will also be a relatively flexible polymer chain with many, many different, the overall chain, the long chain will have many, many different shapes, all with about the same energy and consequently equally accessible. Here's a picture of what this looks like. Now, again, you see a small tendency for the chain to go in a helical form, and I will come back to that point. But this, the helical form in this polymer is much less pronounced than the first one I showed you. This polymer is changing all the time. If you were to close your eyes and come back and look at it a brief time later, it would have changed its shape completely. But nevertheless, there is a small tendency for it to be going in this way. And that has a consequence which I'll come back to at the end of the talk. Okay, now this is really all I'm going to say about random coil polysaccharides. They are probably the predominant, probably more polysaccharides are random coils than any other kind. But they aren't altogether as interesting as some of the other ones, so I want to spend more time talking about other kinds. What I do want to emphasize, though, and I think Professor Cesaro mentioned this, this is all based on computer modeling. You can legitimately ask whether the computer modeling is reliable. Does it have any physical meaning? And one of the things that I've always tried to work on is to make sure that when we use a computer model like this, that we can demonstrate that it has reality, physical reality. The way you do that is use those physical models to calculate properties that are observable properties of the polymer chain, things that you can determine in the experimental laboratory. You saw a picture there from 1967 when Attilio and I were hovering over a machine in the laboratory. That machine was the sort of instrument that you can use to get information, experimental information about size and shapes of macromolecules. And so a strong component of this part of my work has been to demonstrate that there is good agreement between what you can calculate from the theory and what you can observe in the experiment. But once you have the theory, you can probe the system, you can explore the system far beyond the ways that you can see it experimentally. There are limitations to what you can see experimentally. But once you have a theory that you believe in, then you can explore many features, such as the shape like this. You could never, there's no experiment that will ever allow you to see the molecule, freeze the molecule in the shape. There's currently no experiment that will allow you to do that. So the experiments and the theory have to go together in a way that convinces you that the theory is reliable. Okay, now let's talk about rods. I'm going to talk more about this part than anything else. Usually these are what we call homopolymers. That means all of the sugars are the same, or if we have several different sugars in the chain, they form a regularly repeating sequence. So it's A, B, C, A, B, C, A, B, C. So it's this regularity of sequence that leads to the rod-like shape. Typically, the links in rod-like polysaccharide chains are rather inflexible. So we saw 3 examples, one that was extremely flexible, one that was rather flexible, and one that was less flexible. But now for the rod-like chains, the linkages are usually less flexible than in the ones I showed you before. And the thing that characterizes a rod-like polymer is that instead of having a very large number of shapes with very similar energies, now we have a very small number of shapes which are very similar, which have much lower energy than everything else. And so all of the polymer chains in the sample fall into this single hole in the energy map, and they sit there because that's the lowest energy form, and every other form is much higher energy. I'm not going to be talking about that characteristic of these chains right now, but I just want you to understand that these are different because there is one conformation that is energetically preferred over all the others. This is one of the most well-studied of the rod-like polymers. Here, this is the backbone chain, so there are 2 sugars here, and this chain goes on for many thousands of sugar residues. In fact, this chain up here, which is the backbone of this polymer, is just like cellulose. And then there's a side chain consisting of 3 other sugars that comes off. So every second sugar in the main chain is decorated with a side chain that has 3 more sugars. So you can see now that this polymer is becoming really quite complicated. And among other things, there is here a location in which there's electrical charge. A negative charge is situated right here, and another electrical charge situated out here. So this is a polymer that's not only a polymer, but it's also an ion. It's ionic, it's charged, and that has important consequences for its behavior. Now, this is an experiment with this particular polymer, xanthan, which this is a fairly easy experiment to understand. I do want you to understand it, so I'm going to take a little time. Down here along this axis at the bottom, this is the concentration of a solution. So we've dissolved xanthan in water, and as we move from left to right, we're talking about solutions of increasing concentration, higher and higher concentration. What's being measured on the vertical axis here is the viscosity of the solution. Just so how thick is it? How easy is it to move the fluid? And this is a logarithmic scale, so when I move from here to here, I've increased the viscosity by a factor of 10, and by the time I've come up to here, I'm 10 times more. So that's a 100-fold increase in viscosity. Now, the different curves that you see here correspond to different rates at which we are shearing the material. It's not really important to think about that. We can just look at the top curve, which corresponds to shearing the material in a very slow way. The viscosity depends on the shear rate, and you can see over here different shear rates lead to different viscosities. But the real important thing here to see is that, as you might expect, as you increase the amount of polymer dissolved, the viscosity goes up really rapidly. But then something quite remarkable happens. At about this point, say 10% polymer, the viscosity now decreases very abruptly as we add more polymer. So adding more polymer to the solution produces lower viscosity. That's really a quite unexpected behavior and something that one needs to investigate. Ultimately, though, what we find is that at some point, at certain concentrations, say around 12%, it doesn't matter what the shear rate is, and it doesn't matter what the concentration is, the viscosity becomes constant. So you can add more polymer to the system, and you keep having the same viscosity. So this is if you're a polymer chemist, you will recognize this as rather unusual behavior. If you're not a polymer chemist, you may say, what of it? But in any case, it's this is the observation, and in fact, it's quite an unusual observation. So how do we explain this? This is called a phase diagram, and what's plotted along the bottom here is the concentration of the polymer, starting over here with no polymer and going up to 100% polymer. So this is the same quantity that we saw at the bottom of the other slide, and this is the temperature. And the way you understand this diagram is that over here in the upper left where you see the letter I, those are polymer solutions that are isotropic. That is, the polymer molecules, these rod-like molecules, are oriented in all possible ways. There's no preferential orientation. Over here in the region that's labeled A, the polymer molecules tend to be lined up. They're oriented all along the same axis. They're not lined up in a line, but they're parallel to one another, all pointing in approximately the same direction. And then there's a narrow region between, which is the only part of this diagram I'm going to talk about, where we have both phases. We have the isotropic phase and the anisotropic phase mixed together. So that's called the B for biphasic. Now, the experiment we just did starts over here and moves across. So as long as we're in the isotropic region, the viscosity is going to increase as the concentration of the polymer increases. But when we enter this biphasic region, what's happening is that the solution is now, part of the solution is becoming a liquid crystal. Part of it's becoming highly oriented, a liquid crystal. So in this region right here, as we increase the polymer concentration, the proportion of the material which is anisotropic is increasing, and the proportion of the material which is isotropic is decreasing. And by the time we reach this point, the solution is wholly or completely anisotropic. So here is where the viscosity is increasing, here's where it's decreasing, and over here, all the polymer molecules are lined up in the same direction, and they have a viscosity which is dominated by the fact that they're already aligned. Now, let's look at why they align. Here's a kind of simple diagram that helps you understand why this happens. As long as the solution is relatively low concentration, the polymer molecules can be randomly oriented. There's no reason for them to line up. There's plenty of room for the molecules to move around with random orientation. On the other hand, at some point, the system becomes very crowded. Okay, so as long as the polymer concentration is fairly slow, these are the polymer molecules are anisotropic. But here, at some point, some of them have to line up. If you're going to put more polymer in there, the only way you can make it fit is for some of the molecules to line up. So this is the biphasic region where the two phases exist at the same time. And then here is where the concentration is high enough so that all the polymer molecules have to be lined up. You'll see there's a domain here where they're all oriented left and right. Here's another domain where they're oriented up and down. And when you have the system like this, it can flow very easily. The viscosity will be very low because it's very easy for this system to flow because the molecules are already lined up. When you're over here in the isotropic region, the flow is much more difficult. The molecules all have to line up before they can flow. So you have to put a lot more energy into the system. And in here, you have a mixture of the two. So this is an interesting behavior, I think, that it explains, in fact, why this particular molecule, xanthan, is so commercially important. You'll find it, if you look on the label of many foods, you'll see this molecule, xanthan gum. It's used in the food industry a lot. It's used in other industries as well, but it's very useful for controlling texture and feel for foods. And it's because it has great versatility in terms of the the textures that it can convey to the foods that it's added to. Now, I want to stay just a little bit longer on this polymer. I want to do a different experiment here. Let me see if I can go backwards on this. The experiment we talked about was to keep the temperature constant and increase the concentration and move left to right across this diagram. But now I want to do a different experiment. I want to start at a low temperature at some fixed concentration and raise the temperature and pass through this biphasic region by raising the temperature. So it's an alternative way of passing through the biphasic region. So that experiment is here. Now on the bottom we have temperature, and we have viscosity again here, and we have several different concentrations. But let me just focus on two of these concentrations. I'm going to take first the solid dots right here. So this is a fairly low concentration, 2% xanthan. And what you see is that as you raise the temperature, the viscosity goes down. So it's quite as expected. If you raise the temperature, it becomes more fluid, less viscous. But something happens right here, because you'll see that the slope of this decline increases rather abruptly. So right here at about 64 Celsius, the viscosity begins to drop more rapidly. What's going on there is that the helix, or the double-stranded helix, and I forgot to mention that this molecule, the reason it's so stiff is because it's two polymer chains wrapped around one another. They are very much like the DNA double helix or the nucleic acid double helix. So this chain is stiff and rod-like because it's double helical. And right at this point, at 64 degrees, that double helix starts to unwind and it comes apart. And so instead of being a rod-like double helix or a very stiff double helix, it's now becomes separated chains which are much more flexible, and they have lower viscosity. So as you move down here, you're moving to, you're having this chain melt and become totally random coils. Now, along this curve, there's everything. It started off in the isotropic region and it stayed in the isotropic region. We never pass, there's no liquid crystals along this curve. But now what happens is we're going to cool this system down. So we don't return along this curve. What happens is we now return along a curve here in which the viscosity goes up by over a factor of 10. So now these random coils that at the beginning had been paired together as double helices, double strands, now they can't find their partner, and what's formed is a network. So as we cool, a network is being formed, one that has much higher viscosity than where we started. This is another illustration of the versatility of this polymer in commerce. Let me just look at one more of these. Let's look at the curve down here at the bottom. This is a curve that starts at low temperature. It's already fully anisotropic. So whereas this one was isotropic, the one I'm talking about now is anisotropic, or liquid crystalline. And what happens here is we heat it. You see a very small decline in the viscosity. And now, as I raise the temperature, the viscosity goes up. So that, again, is quite unusual behavior. That's raising the temperature of a fluid, its viscosity should go up. The reason the viscosity is going up is that as we move from this point right here to this point up here, we're passing through that biphasic region. We're converting the liquid crystalline form back into the isotropic form. So the isotropic form is much more viscous than the liquid crystalline form, and even though the temperature here is higher than it was here, the viscosity is higher because now the polymer chain has been converted to an isotropic form. As we heat further, we get to the point where the double helix begins to melt, right about here at, say, it looks like it's somewhere around 70 Celsius, and we then return, as the temperature is reduced, to this gel-like state. So the point of this discussion is just to show you that this is a very versatile polymer, and the applications of this polymer in commerce depend very much on the concentration that you're using it at, depend very much on the thermal history. What sort of cycling of the temperature has happened to this it's actually put into use. Okay, let's see what's next. This is just to show you that the experiments that we just talked about, in which we change the temperature and observe the viscosity, is actually a way to discover experimentally the very narrow region of the phase diagram. So if you look, for example, at these two curves right here, these two parallel lines that are labeled 0.1 molar sodium chloride, that's the part of that phase diagram that's biphasic. And so this is an experiment that the phase diagram I showed you was very schematic. It wasn't based on an experiment, it was simply based on a theory. Now we see that we have an experiment that can confirm that the phase diagram does in fact look just as we expected it to. Okay, so I think I'm going to pass over the next couple of slides and go on to talk now about rings. What we mean by a polymer ring or a polymer circle is usually we just have a polymer chain that's connected together, chemical bonds, and it just makes a circle. So it's easy to understand how that can happen. Instead of connecting one end of the chain to another unit, you just bring it around and connect it to the other end. So typical polymer rings are covalently linked. And there are a number of polysaccharides that are like that, and they're very interesting to look at, very interesting to study, but I'm going to talk about a completely different kind of polymer ring. The other thing that you ought to notice is that as a polymer chain is growing, it's much easier for it to grow in a linear way than for it to grow in a cyclic way. Because for it to grow in a cyclic way, this end of the polymer chain has to find this end. And that's quite unlikely. That is, this end can fly all around relative to this one. And it's a very unlikely event, low probability event for two ends to come together. So the study of polymer rings it brings in a completely new consideration of figuring out what the probability is that the ends will meet. Now, I'm going to show you about a kind of polymer ring, though, that isn't covalently linked. It makes the ring by a completely different mechanism. Wrong button. Let me go on one. This is the structure, the basic structure of another kind of polysaccharide. And here we have the backbone. This is just a very small segment of the backbone. So this backbone runs on very, very many sugar units. These are glucose again. And then every third one, we have a side chain. This is a very characteristic motif of the microbial and bacterial polysaccharides. You saw that again in xanthan. Xanthan had a backbone. Every second sugar had a 3-sugar side chain hanging off of it. Here we have every third backbone unit has a single sugar side chain hanging off of it. Now, the linkage along here corresponds to when I was talking at the beginning about ropes or filamenti. This was the last example I talked about, and it's a chain that has a weak tendency to form a helix, but it's not a very strong tendency. But it's very interesting that this chain tends to exist in nature as a triple helix. So whereas xanthan was a double helix, this one is a triple helix. And we have here, it's a little hard to see in this particular diagram, but there are 3 chains wrapped around a common axis. And they are, so you can imagine this is extremely stiff because there are 3 chains all wrapped together. This is looking down from the top. And what you see here in the center is a representation of the very strong interactions that are holding this triple helix together. Now, this is a very, very stable triple helix. And we don't want to talk here about the details of this bonding on the interior, but it's very strong. And so if you want, you can dissolve this material in water, and you can get very high concentrations. It's surprising how easily it's dissolved in water. And if you want to raise the temperature now and disrupt this helix, Remember that the xanthan helix was disrupted by raising the temperature somewhere around 60 or 70 degrees. You have to raise the temperature of this helix to well over the boiling point of water to disrupt it. And that's the consequence of these very strong interior interactions which stabilize it. So this is the basic chain. And now this is another phase diagram. We don't have to pay much attention to this. We just need to look at the left-hand side here. This is scleroglucan dissolved in pure water. And what we see is that at this point, about 130 Celsius, there's some sort of change. That is, this boundary up here signifies the fact that the chain is passing from the triple helical rod into a disrupted helix. And so above 130 degrees, you have random coil polymer chains. Below 130 degrees, this molecule assembles into triple helical rods. So I want to describe an experiment for you in a minute, which involves crossing this boundary, first going up and crossing it that way, and then looking to see what happens when you come back down. So if you start with triple helical rods and disrupt them by raising the temperature, and then cool the temperature back, do you get the rods back? So that's the question. This is the experimental technique we're going to use. Attilio showed you some pictures like this. This is an atomic force microscopy experiment. This bar here is 400 nanometers. That tells you what the scale is. So you can see that these polymer chains, some of these are 1,000 nanometers, a micron long. They're very big. They're almost macroscopic. And you can also see that they're quite stiff. You'll notice how extended they are. This little bar here just shows you that with this particular technique called atomic force microscopy, you can measure some of the properties of these chains. So you can measure the length from one end to the other, so it's easy to measure the length. And the other thing that you can measure is the thickness. So down here at the bottom, you'll see this trace. You're going along essentially zero thickness, and then you rise quickly to about 9 angstroms or 1 nanometer, and then it comes back down. So this peak here corresponds to crossing that chain. So with this technique, you can measure chain lengths, you can measure the distribution of chain lengths, and you can measure what's most important for this particular exercise here is you can measure the thickness of the chains. Okay, so now I'm going to take some much shorter chains than these. These are very long. These are pretty much the way you get this polymer from its original source, so it's quite high molecular weight. But in the picture shown here, you're looking at the same polymer, but now you see that the chains are much shorter. And that's because we purposely went in with our scissors and cut them up into smaller segments. So here you have a 200 nanometer bar down here, so you can see here's a chain that's 200 nanometers, here's one that's 150, here's one that's 75. So these chains are somewhere around 200 to 300 nanometers long, or 75 nanometers to 300 nanometers long. And now what I'm going to do with these chains, this is a solution, this is prepared from a solution in water at room temperature. And now I'm going to take this very same sample, raise the temperature up to about 165 Celsius, let it stay there for a few minutes, and cool the temperature back down so that we pass into that region where the rods are again stable. At 165, we expect the random coils to exist. We cool back down. Now we hope that the rods will reform, or we ask, will the rods form again? And what we see is, I think this is very similar to a picture that Attilio had, maybe not identical. But now what you see are, down here at the bottom, you'll see some rods that look very much like the ones we started with, quite a few of them. But then we see something completely different. We see things that we call rings. These are cyclic versions of the same polymer. Now, of course, just by looking at this picture, you can't tell whether these rods down here have the same structure as the ones we started with, and you can't tell whether these circles have a triple helical structure. But because we can measure the thickness of these things, it's very easy for us to convince ourselves that these rod-like ones, the ones that return as rod-like, they have all the same characteristics as the molecules we had before we heated them. So they have the same thickness, they have the same distribution of lengths. So they appear to be reconstituted rod-like molecules. On the other hand, what about these? These are very surprising, because if these are also triple helical, that's like taking this pointer, which is very stiff indeed, and moving it into a circle. So we have to try to understand, how can something that is really very stiff, I mean, these 200 nanometer chains they're almost as stiff as this thing. They bend a little bit, but they're not very flexible. So how can you possibly have them come around and make a circle without being extremely high energy? So that was we were very surprised to get this picture. We didn't expect this at all. And once we got it, it gave us 5 years of work. So here's the way we understand what's going on here. Now, this is just a cartoon or a diagram. And on the left-hand side, we have the rod-like forms. And on the right-hand side, we have the circular forms. And I want you to really understand this, if possible. So down here, we have a single-stranded molecule that's stretched out. It looks like a rod. But two of those could get together, and these little bars here, they just represent the interactions that are holding those two chains together. So here we have some kind of a double-stranded entity. It's not fully aligned, but you could imagine moving the top one relative to the bottom one so that these two came into alignment, and you had a large number of stabilizing interactions. Here's a chain. Here's an entity or a structure or a cluster that has 3 chains in it connected together, and the way they're connected here, there's no triple-stranded structure, but up here we imagine pushing on the ends until they start to overlap, and so these dark lines, in this particular representation, these dark lines correspond to the triple-stranded helical regions. The lighter lines correspond to the double-stranded regions. And you can push on the 2 ends of this until you get a fully wrapped up triple helix. So that's the model of the triple helix that we're talking about. But the things that we see in the micrograph could be these, they could be these, they even could be these. That is, it's not real easy to tell these apart. You can tell this kind from that kind, but how do you tell these two apart? They're pretty similar, and the resolution that you get with the microscopy makes it difficult to distinguish these two. And then you have to appreciate that there could also be much larger clusters. You aren't restricted to having just 3 chains together. Why not put 4? Why not put 5? So we have other possibilities here. So these are our model ideas of what the linear structures look like. Now, what do the circular structures look like? Well, here is a hypothetical perfect circular triple helix. So it's 3 chains, and they're not covalently linked. So let me say aside, one of the things that you have to understand about these diagrams is that because we've taken 3 dimensions and pushed it into 2 dimensions, there's some distortions here. So each of these chains should be understood as having the same physical length. They don't look the same length here because you can't draw it that way. But if you think of these chains as having the same physical length, then we have 3 chains wrapped together, and they make a circle. And so let's look at this center chain. So here's where there's no covalent linkage, but the gap in that one is sealed by this chain here, which covers that gap, and this gap in the second chain is covered by that. So you can easily construct a triple helix that's circular, although you must ask, why would this happen? Because this form must have much higher energy than this form. We know that this form over here is the low energy form. So why would this happen? Well, before we talk about why it happens, let's just notice that in addition to having these perfect triple helices that are circular, you could have some less perfect ones. That is, you could have 3 chains hooked together here in such a way that there was one region of single strand on the circumference, another region of double strand on the circumference, and a couple of regions of triple strand on the circumference. So there's lots of ways to build these circles, and they don't all have to be perfect. Another thing that's really, you really have, it sort of hits you in the head, you don't have to have 3 to make a triple helix. In fact, you can take one chain, one of these guys right here, and you can wrap it around itself 3 times, and so with one chain you can get a triple helix. if it's circular. Now, I know this one looks a lot longer, but again, that's a distortion of the representation. They're really not any longer, so I'm taking a chain like this and I'm wrapping it around itself 3 times until I have a triple helix. So that's, we call that a U sub C, that's a unimer, 1 chain, circular, cyclic. Here are 2 chains, cyclic. Here are 2 chains that are cyclic and fully triple helical. So this is the array of species that we postulate are being observed in the micrograph. Now, what sort of proof do we have that this is a proper picture? Well, we don't have absolute proof. This particular diagram was part of a theory that we developed. So we developed a statistical mechanical theory that attempted to explain what's going on here. It worked pretty well, wasn't perfect, but I learned a lot from it. So what we observed was that in these micrographs, let me just back up one here. When we examine these micrographs in detail, we find that the rods, on the average, the rods have a longer length compared to the circumference of the circle. So the circles, if you measure the distance around the circle, on average, is less than the length of the rods. You can't see that in this particular picture, but my student actually measured thousands of molecules. I mean, so there's good statistics. It wasn't just 2 or 3 molecules, it was thousands of molecules. And so on the average, the circles are shorter than the linears, and the circles are thinner. That is, they aren't as high above the plane of the micrograph. So those observations need to be explained. And that's the last thing I'm going to talk about. First, let's just understand why it is the circles form at all. Why should they form? They're very high energy. Well, the thing that enters here is a concept called degeneracy, or multiplicity. If you look at this form right here, you'll see there's a gap. In this inner circle, there's a gap. Now, this gap out here could be anywhere on this circumference. And the molecule would look the same in the micrograph. It would have the same energy. Likewise, this gap here could be anywhere around the outside. So there are very, very many ways to build this triple helix because the relative locations of these gaps can be anywhere on the circumference. So that's what we call degeneracy. Many, many ways to do it. On the other hand, there's only one way to build this one. There's only one way to build a perfect linear triple helix. So this one is favored simply because there's so many ways to do it. Actually, they occur at about 50-50. So what the degeneracy here is just about enough to compensate for the higher energy. And so now we're talking about something that comes into thermodynamics. We're not just talking about energy, we're talking about the entropy. So you can say that this one makes more contribution. This one has higher entropy than this one does, and that's why this one, when you have collections of these, that's why this one happens at all. Finally, why are the circles thinner and why are they shorter? Well, you might think that this kind of circle right here would be favored over this kind because whereas here you've got to bend a triple helix to make the thing close, here you've got a region of single strand which is very flexible. So closing this ring in the presence of part of a single strand on the circumference should be much easier to do than this. So we speculate that a lot of the rings actually look like this rather than like this, because this is definitely a high energy form. In our theoretical calculations, we don't find very many of these. We find a lot of these because these can be formed without so much cost and energy. But if most of the circles look like this, why is the circumference smaller than this length right here? Remember, this length on average turns out to be larger than this length, but it looks like this should be longer. The reason for that is because we think a lot of the circles may be of this type or this type. Think about this type. You start with a single strand, you start wrapping it on itself. You've wrapped it around once, you've got a double strand. Wrap it around again until you've got a triple strand. And now the circumference of this thing is just 1/3 of this. So that if the circular samples are in part incompletely helical or consists of dimers or unimers, then you can explain why these circles are both thinner and shorter. Okay, so that's where I'm going to stop, except for just giving credit to the people who did almost all of this work. On the left, you see the list of people. I listed here only the people who were in my laboratory or with whom I collaborated very directly. There are a lot of other people who are authors on papers that I've written, but whose contributions were less important. So almost all the people in this list right here are my students or my postdocs, and there are a few people who are professional collaborators. So here you see Attilio Cesaro occurring relatively early in the list because he came to my lab very soon after I started my work there. Farther down on this list, you'll see Ranieri Urbani, who came to my lab. Here are a couple of guys that I collaborated with recently. They're from Genoa. And then this is the group of people who worked on the rod-like material. And the work that I talked about today, the viscosity work, was all done by Hucheng Li. And then the work that I did on the rings, you'll see there weren't very many collaborators because most polysaccharides and most of the ones I've studied have this form. Some of them have this form, and some of the most interesting ones certainly have this form. Notice here Gaio Paradossi, another Italian who came to my lab in about 1980, and he's the person who actually discovered that xanthan is a double-stranded helix. It took us about 10 years to convince anybody else, but after about 10 years, people began to believe it, and he's now at University of Rome, number 2. And then not so many people work on rings because rings are much more rare. Okay, that's the end.
Thank you, Professor Brant, for your superb lecture. And now I will invite my colleague, Professor Benedetti, to present you the addition of carbohydrate research that is dedicated to your career and your activity as a scientist.
L'ultima parte di questa cerimonia è la consegna al professor Brant di un volume speciale della rivista Carbohydrate Research, che l'editorial board di questa prestigiosa rivista di ambito chimico hanno deciso di dedicare al professor Brant in considerazione del contributo da lui dato al progresso degli studi e delle conoscenze nel campo della chimica dei carboidrati. Il professor Horton, che è l'editor di Carbohydrate Research, non ha potuto essere qui con noi oggi per consegnare personalmente una copia del volume al professor Brant, ma Attilio Cesaro, che è coeditor di questo volume speciale, mi ha chiesto di essere il tramite in questa cerimonia, cosa che io mi accingo a fare con grande piacere, ma desidero prima leggervi e leggere al professor Brant un messaggio del professor Horton. I will now read a message from Professor Derek Horton, editor of Carbohydrate Research. On behalf of the editors and publishers of Carbohydrate Research, I would like to transmit on this occasion sincere good wishes to our distinguished colleague David Brant, expressing high appreciation for his contributions to the field of carbohydrate science and especially for his originality and insight into the physicochemical aspects of carbohydrate structure. Many friends and colleagues from around the world have joined together to recognize the leadership and example of David Brant by contributing original research papers grouped around the theme of conformations of oligo and polysaccharides, and here gathered in an outstanding special issue of the journal, prepared under the editorship of Attilio Cesaro and myself, Derek Horton. It is a particular pleasure and privilege for Attilio and myself to have this opportunity to present to David a special souvenir copy of this issue. We have greatly enjoyed working during past months with the contributors to this collection. We have also appreciated the valuable work of many reviewers who have assisted in evaluating the manuscripts and helping the authors to present their work at the high scientific standard that characterizes David Brant's sound published research. I regret that I'm not able to be present personally on this occasion to join with friends and colleagues in celebrating David Brant's achievements, but nevertheless wish him well and all success in his future endeavors. Derek Horton, Editor, Carbohydrate Research. So, I would like to add
Bene, con questa parte conclusiva la cerimonia si chiude. Noi siamo grati naturalmente al professor Brant per la sua lezione, per la sua presenza qui. Sono grato a tutti i colleghi e agli studenti e adesso c'è un brindisi per chi vuole scambiare anche due parole con il professor Brant giù nella sala atti della Facoltà di Scienze Politiche che gentilmente ci è stata messa a nostra disposizione.